Thank you, Miles. Welcome, everyone. I'm really happy that you're here with us today for this Cell and Gene Live. In just a minute, we're going to dive into our presentation featuring expert panelists J. Andrew Case, he goes by Andy, head of cell supply chain, cell and gene therapies at Genentech Roche, and Daniel Getts. He is the CEO and co-founder at Myeloid Therapeutics. I really encourage you to submit questions for our experts as you think of them via the Q&A tool. We will get to your questions as part of the presentation, so please do submit them. We'd love to hear from you. And finally, I too would like to thank Thermo Fisher Scientific for partnering with us for today's event. All right, so given the evidence supporting the effectiveness and safety of clinically approved mRNA vaccines and coupled with the growing interest in mRNA-based therapeutics, mRNA technology is really poised to become one of the major pillars in cancer drug development. There are so many facets to this conversation, so we're going to jump right in. And we're going to start off with Daniel. And my Cell and Gene Live events, I love to level set the conversation. And so, Daniel, I'd like to start with you. Please give us a brief overview of the different approaches being used to leverage mRNA technology in the cell and gene therapy space. In addition, I'd like to hear how Myeloid uses mRNA to program cells as perhaps an alternative pathway to manufacturing a CAR therapy. So can you give us a little bit of background?
Yeah, fantastic. I will do that. Thank you for the opportunity to be here. First, I'll start with Myeloid before I take a broader approach. Myeloid, we founded the company back in 2019 with Ron Vale and Sid Mukherjee, with a focus on how we can use the myeloid compartment, the orchestrator of all immunity, to really tackle high unmet medical needs and indications like solid tumors, where current approaches really haven't got us to where we really want to go. And what's quite unique about the approach that we were using is myeloid cells include things like dendritic cells, macrophages, neutrophils, and so on. Traditional approaches like viral-based vectors, had a tendency to drive everything to be a macrophage when we use them, or just killed the cell. And so RNA was something that we used mostly because biology, and science pushed us there. And at the time, I don't think we truly appreciated the versatility and the amazing capabilities of RNA. But since then, we've been able to use RNA, and we've built a set of next generation technologies, including in vivo mRNA CAR delivery, where I believe we're the first in the world to deliver a CAR using RNA into human beings. In this case, patients that have TROP2 expressing epithelial tumors. And we've also used RNA to ex vivo engineer myeloid cells. And in this case, we take a leukopak, we isolate the myeloid cells, and we use RNA to deliver a CAR. And what I can tell you from the cell therapy and from our in vivo delivery technology is we are able to engineer these cells with significant efficiency using RNA. In our cell therapy, it's a one-day manufacturing process. We don't have to activate the cells or culture them. And in vivo, myeloid cells, because of their job naturally, which is to pick up things that shouldn't be there, like lipid nanoparticles. We've seen in non-human primates up to 40% of the circulating myeloid cell compartment expressing the CAR after delivery of the product. So, the approaches that we use show broad immune responses after treatment in non-human primates, animal models, and in humans. In the case of our cell therapy, we've seen in one patient, in one tumor, 25 new TCRs 17 days after treatment. So, we have a very strong belief that, this approach is taking a significant step forward, and, bold vision that using RNA, we can accelerate not only our technology, but, a number of other technologies as well. And you opened the question really more of a broad landscape, kind of component to the question. And I think it's important to point out that RNA has the potential to really transform across a number of different areas. Obviously, mRNA vaccines is the tip of the spear. But we're starting to see approaches with aptamers, self-amplifying and activating RNA, circular RNA, microRNA, antisense oligonucleotides, CRISPR gene editing, CAR T cells, so on and so forth, across not only rare, genetic disorders and oncology, but respiratory diseases and so on and so forth. And I think even, something I recently was exposed to was the potential for using RNA in the GLP space. And you can imagine with obesity and so on and so forth really taking off right now that there's, a significant opportunity there. And I think one of the things about RNA, obviously delivery and stability is something we're going to discuss today. But if we think of it in the context of cost of goods relative to other approaches, it's actually very, very, I think, ahead of the curve, in an environment where there's a lot of pressure on cost. And I think the other element about RNA, especially what we've experienced at Myeloid, is the versatility and rapidity in which you can test a number of different constructs. If you think of the cell and gene editing or delivery space, folks using lentivirus, you've got to generate your plasmids, you've got to generate your lentivirus, and then if you're using a cell therapy, make the cell therapy. This can take, depending on how good your process is, 30 days to 90 days on a good day. In the context of RNA, especially if you have your platform up and running, you can generate new constructs in 3 to 5 days to test. So, I really think that the versatility of RNA and where it can be applied is limitless. In the context of Myeloid Therapeutics, obviously we're an oncology company looking to deliver and modify immune responses in cancer patients. But our belief in RNA as a whole has really driven us to, partner with larger CDMOs. And we're working with the government actually in Australia to build a facility, GMP manufacturing facility there that will help support as we're moving forward. So hopefully that gives a rounded view of, not only where RNA can be used, what we're doing at Myeloid, but really, to me, I think this is a huge opportunity, and I don't see RNA going anywhere soon. I think it's just going to continue to expand.
Oh, 100% agreed. So thank you for that background. And as you mentioned, we're going to get into stability, cost of goods, supply chain, all that good stuff. Before we do, Andy, I want to talk a little bit about Genentech's joint venture with BioNTech that's focused on autologous mRNA cancer vaccines that are, custom made for each patient. You've been in the news this year quite a bit, and I certainly don't want to steal your thunder. So please bring us up to speed on where we're at with that.
Yeah, sure. So thanks for the opportunity to be here. And just to level set, I'm definitely going to bring the perspective more of how we operationalize these therapies. And I have a very limited understanding of the science behind the therapies. I'll leave that to Daniel to explain. And as he mentioned, one of the therapeutic areas, one of the tips of the spear, as he said, is leveraging RNA technology, the vaccine side of mRNA technology, to create essentially a custom cancer vaccine for each individual patient. So it's in a sense an autologous therapy because it starts with the patient's tumor cells that are sequenced to identify new antigen targets, and then these targets are programmed into the RNA to create a vaccine, with the belief that this will create an immune response or train the immune system of the actual patient to attack the specific nature of the cancer that they have. And we're actively in clinical trials trying to prove this out as we speak.